start the recording. So thank you everyone for joining the inaugural ACHV discussion panel event, where we're going to talk about how medical science has contributed to the improvement of people's lives for people living with hepatitis B and hepatitis C in particular. My name's Thomas Tu. I'm the president of the Australian Centre for Hepatitis Virology, which is the National Society of Medical Scientists that research viral hepatitis. I'm a research scientist myself, and I've studied the molecular biology of the hepatitis B virus for the last 14 years. I also live with chronic hepatitis B, so I have a stake in all of what we're going to be talking about today. I'd like to acknowledge that I'm speaking to you from the land of the Darug people, the traditional owners and custodians of this land. I would formally like to welcome and pay my respects to their elders past, present and emerging. I would also like to acknowledge all Aboriginal and Torres Strait Islanders here today and pay my respects to your elders past, present and emerging. I recognise that viral hepatitis, along with many other health conditions, disproportionately affect your communities, and that this is an issue exacerbated by past and continuing injustices and inequities that are built in our society. I think all of us must take some responsibility to know what has come before us, how it's resulted in what's happening today, and how and where it's leading us into the future. And indeed, that's our major theme of tonight. So before I introduce our highly experienced and wonderful expert panel who will be discussing this theme, I'd just like to make some housekeeping announcements. So the structure tonight will be first a panel discussion introducing the background of viral hepatitis and sort of each panelist's impression about how lives have improved because of scientific discoveries we've made over the last few decades. Please be aware that we're recording this session. And so this is because others who are not in our time zone should be able to watch this session at least, if not participate. So the main part after this discussion and the bit that I hope you all will participate in is the Q&A session. And hopefully we'll have about 45 minutes to answer all the questions you have out there. Now, we've muted everyone for the time being, so no one embarrasses themselves by having kids screaming in the background or accidentally going to the toilet with their mic on. But if you'd like to ask a question about something, please use our Slido link and type it in there. We will make that URL available in the chat right now. And so you can go there and ask your questions. So during the Q&A session, my co-host, Dr. Chaturaka Rodrigo, will offer you a chance to read your question if you put your name next to your question. So you have the chance to read it out to the panel, or you may leave it to him to read the question out for you. He'll also be reading some pre-submitted questions from the affected community. Aside from this, Dr. Rodrigo is the secretary of ACHV and leads a research group at the Kirby Institute at the University of New South Wales. There, his work concentrates on how our immune systems react to viruses and how those viruses evolve to get around them. He's studied all sorts of viruses, including dengue viruses, coronaviruses, and importantly here, hepatitis C virus. Also on our panel, we have Professor Heidi Drummer, who is the past president of ACHV. She's the program director for disease elimination at the Westmead Institute in Melbourne and the scientific director of Burnett's diagnostic initiative. Her scientific and advocacy work has focused on trying to develop a vaccine that can protect people from hepatitis C, something that we still don't have. She has a wealth of knowledge, having been in this research space for over 30 years. So she'll be answering your questions on hepatitis C virology and immunity. We also have Professor Scott Bowden, who I'm very grateful could be convinced to come out of retirement for this event. He is the former head of the Vigil Molecular Microbiology Laboratory at the Doherty Institute in Victoria, and a senior scientist in the WHO Regional Reference Laboratory for Hepatitis B. He is one of the top guys in Australia and indeed the world when it comes to testing for viral hepatitis in the blood and how that relates to the clinic and the science. Indeed, he's consulted for the government numerous times about this and telling them, you know, what we should be doing, which of course they always do, right?
Yeah.
He'll be the one answering questions on the particulars of lab results, testing policies, and diagnostic technologies. We also have Jennifer McLaughlin, who's also from Victoria in the Doherty Institute. Jen is a senior epidemiologist for the WHO Collaborating Centre for Viral Hepatitis. She's the head of a great project called the National Viral Hepatitis Mapping Project, where her team uses health and social databases to look at who and in which regions in Australia are most affected by viral hepatitis, and also modelling how best to eliminate hepatitis on a national and global scale. She'll be answering questions on the much bigger picture of medical discoveries and its impact on health. Finally, last but not least, we circle back to New South Wales. Associate Professor Simon Strasser is a senior staff specialist in the A.W. Morrow Gastroenterology and Liver Centre and the National Liver Transplant Unit at the Royal Prince Alfred Hospital. There, she is also the Director of Hepatology Clinical Trials, which does include the ones for hepatitis B. Among many other things, she is the President of the Gastroenterological Society of Australia, which is the national body for all the liver specialists in Australia. So she has decades of experience as a doctor in treating people with viral hepatitis and organising liver transplants if it all goes south. So she'll be handling the more clinical questions on viral hepatitis for the panel. So Simon, let's start the discussion with you. Can you tell us what viral hepatitis is? You know, if someone has hepatitis B or C, how do you know? What do they feel? Is hepatitis C worse than B or something like that?
It's a really interesting question, Thomas. So I'm just going to answer that from a clinical perspective, obviously. So in fact, the vast majority of people we would see with viral hepatitis, whether it be hepatitis C or hepatitis B, are asymptomatic and they've been picked up on a blood test. And the reason they've had a blood test is either because their doctor or the nurse they're seeing or a healthcare professional feels that they've got an increased risk for having hepatitis B or hepatitis C because of identified risk factors, such as where they were born, procedures they might have had, a history of perhaps injecting, you know, depending on which virus we're talking about, or they've come forward for testing because they've identified they might be at risk, or they've got a family member who might have viral hepatitis, or they've been found to have abnormal liver tests just on a routine blood test. We still, though, even now, still see people come through the door with liver failure or liver cancer where the first time they get diagnosed with hepatitis is when they're presented with really those end stage manifestations. And so while that's unusual, it's really devastating when that happens, because, you know, what we have seen with the advances over the last couple of decades in the treatment for hepatitis B and hepatitis C is we've seen amazing, you know, turnaround in terms of advanced liver disease. When we see people present with advanced disease and they've never been diagnosed before, we've lost that opportunity, and that's really, really devastating. Which is worse? People ask me that. They're both, you know, if you've got cirrhosis, liver failure, liver cancer, it doesn't matter which virus it was due to, they're both bad. And now they're both eminently treatable. If we diagnose it early, particularly. So I don't think one's worse than the other. They both cause chronic liver disease, and they both have devastating consequences if undiagnosed and untreated.
Yeah, thanks, Simone. So I guess to find out for sure if someone has hepatitis B or hepatitis C, we need to send that blood test to the pathology lab. So Scott, you've worked for the regional WHO reference lab in Australia. So what is a reference lab, and how does it help us find out whether someone has viral hepatitis B or hepatitis C, and how we treat it?
I think, Thomas, in that role, a reference lab, the other laboratories have caught up. But in the early days, we really just had hepatitis B surface antigen and surface antibody and core antibody. There were no hepatitis C tests. In fact, hepatitis C was called non-A, non-B because it wasn't hepatitis A or hepatitis B, but still caused hepatitis. So one of our early roles was trying to develop tests that would help out. So we were like the first lab in Australia to set up hepatitis B DNA testing. And with some of the co-workers, so I've seen some have joined the meeting, we set up Hep B genotyping, we set up hepatitis B drug resistance testing. And so once the antibody...
There, sorry, what do you mean by genotypes?
Right, so hepatitis B, there's got lots of strains, for want of a better word. You can look at the genetic material of the virus, and they can be divided up into alphabetically for hepatitis B, A, B, C, D, E, F, G, H,
which is very confusing,
etc. But really the common ones are A, B, C, and D, and there again can be subdivided. So there's a couple of A's, there's several B's, there's lots of C's, and there's a few D's, and they're subtly different, and they can cause disease in subtly different ways. Some are more likely to be associated with cancer, for example, with liver cancer. So that's quite important to be able to do that. But now there are commercial tests available which will help you out with those. And hepatitis C, for example, originally we didn't have a genetic test to look for the virus, the PCR, which now, because of COVID, everybody sort of knows what PCR testing is now. So we sort of set up a PCR testing where we had samples coming from all over Australia. Again, hepatitis C genotyping was important in the early days of treatment, but the hepatitis C genotypes were numerical, 1, 2, 3, 4, 5, 6. As part of the reference laboratory, we also look after the Western Pacific region, so we would have quality controlled reagents going out to those places, and we would look at educating the people in different parts, China, Malaysia, Singapore, the Pacific Islands. People from New Zealand would come over and pick up these techniques so they could take them back to our labs, and also look at ways of being able to examine blood samples, which were difficult to procure going through normal channels, but look at things like dried blood spots being sent to be able to examine whether hepatitis B or hepatitis C is prolific.
So I guess there'd be reference labs for different regions all across the world. Yep. And I guess one of the points might be to make sure that everyone's on the same page when it comes to saying, you know, for this value you have, that means this test is positive. Is that sort of right?
Yeah, well, the WHO is pretty good at coordinating the different regions. Fortunately or unfortunately, depending on how you look at it, the burden of hepatitis B falls mostly in our region because we sort of have China as part of it. So it's quite important, but WHO are aware of it, and there's strategies, there's group meetings of the different regional reference laboratories to discuss these items. Yeah.
Yeah, thanks, Scott. So So in Australia, when someone's diagnosed with hepatitis B or hepatitis C, these are both notifiable diseases when they're positive. So once someone tests positive for hepatitis B or hepatitis C, they get counted and submitted to a central database. And this lets us know how many people have it, right? So from this, we can get the figure of about 230,000 people with hepatitis B and 130,000 people with hepatitis C in Australia. So Jen, you lead the hepatitis mapping project as the senior epidemiologist. Your job is to get that number, I guess, amongst other things. And so I'm assuming that getting that number is a bit more complicated than just counting the notifications that come through. So can you tell us a bit about how you get to these sort of numbers?
Yeah, absolutely. It's a really great question. And I think I was thinking back on this, actually. I feel like what we sort of did is we got the number and then we've spent the next few years making it a better number, making sure that it's more accurate and more detailed. So yeah, it's absolutely a process. So first of all, I just want to give credit to our colleagues at the Kirby Institute, who are the ones who actually generate that number that you mentioned, the 130,000 people living with hepatitis C. And our team here at the Doherty estimates the data around hepatitis B. But both of those numbers are estimated using mathematical models, and both of those models incorporate those case numbers you mentioned, but just in different ways. So for hepatitis C, you're absolutely right. The basis of it is really those notified cases. So hepatitis C has been notifiable in Australia since 1995. And so it's estimated that most people who have Hep C currently have at some point been notified, and we've recorded that case. And so that number is taken and then adjusted to actually account for the total number of people who are currently living with hepatitis C. So that includes taking out the people who have had spontaneous clearance, who tested antibody positive but don't actually have chronic hepatitis C infection. It means taking out people who sadly have died of hepatitis C, also people who have migrated away or who've migrated to Australia. And then it generates an end number of people who we think currently have Hep C. And obviously since 2016, that number has now constantly been updated to account for the number of people who've been cured. And so people have been taken out of that number every year, which has been really exciting to see that number continually going down. So from about 190,000 in 2016 to about 130,000 at the end of 2018. So it's been really great. And then so for hepatitis B, we approach it slightly differently. And so Simone mentioned this when she was talking about why someone might be tested. And because for hepatitis B in globally and also in Australia, the prevalence of Hep B is sort of strongly determined by people's background, and particularly cultural and linguistic diversity, someone's Indigenous status, or their country of birth where they were born. And so for hepatitis B, we use those sort of measures and we collate a model based on those sort of factors. And so migration is a really key input into the model. But then we also look at things like vaccination, which has had a really big impact on Hep B over the years. We look at things like the natural history progression. We look at where people are in terms of the phase of infection. I'm sure we'll talk more about that later. And that sort of thing. And then we also look at births and mortality and that kind of stuff. And so we're able to generate an overall number of people living with Hep B. So that's obviously a bit of a simplification. That's it's a fiendishly complicated process, and I don't do it. I'm not a modeler. We have a fantastic modeler in our team who does all that work. But it does allow us to get a really detailed picture of not just the overall number, so the number of people living with Hep B, but also things like how many of those people with Hep B have cirrhosis, for example, what age distribution of people living with Hep B, how many of them come from particular backgrounds, how many of them are Aboriginal and Torres Strait Islander, or how many of them are born in a particular country. And it also allows us to do things like project what the future number might be, especially given the impact of vaccination and also of treatment. So it's something that allows us to measure those sorts of things. But yeah, that's kind of some of the methods of how we generate those numbers.
Yeah, that's really great. And I think as a researcher, it really helps to know what the target we're aiming towards is, and what what we need to keep in mind. If we develop a cure, where's it actually heading? Is it going to treat the people who need to be treated?
Yeah, absolutely.
So Simone, I guess one of the things I wanted to talk about today was basically how far we've come in trying to treat viral hepatitis. So I want to go back to the year I was born, because I'm quite egotistical like that, I guess. So I was born in 1986, 35 years ago, let's say one generation. So if someone back then came into the clinic with viral hepatitis, what could we do about it? How could we treat it?
Well, pretty much not much. 1986. So I'm a bit older than you. So I was a resident, and so I had 2 years out of university in 1986. So I was seeing people in the hospital largely, and then I was seeing patients with liver disease because I was already doing my training to be a gastroenterologist and hepatologist by 1989, so only a few years after that. And really at that point, we had no testing for hepatitis C. As Scott mentioned earlier, we didn't know who had hepatitis C. We were kind of vaguely aware of this entity of non-A, non-B hepatitis, and that was largely a sort of hepatitis that we knew was there in somebody who might have had a blood transfusion was the main risk factor that we sort of were aware of at that time. And with hepatitis B, we could diagnose hepatitis B with a hepatitis B surface antigen, but there were no therapies. So it was it even predated our horrible treatments of interferon, which really only started to be used by the late 1980s in non-A, non-B hepatitis, and hepatitis B later, I think, than that. And that was standard interferon that really we poisoned people with, but didn't really work very effectively. So there wasn't really much we could do. And then we were starting to do liver transplants for people with liver failure for whatever cause starting in about the mid-1980s, the very early liver transplants. And really, I was working in liver transplant by 1991 and recall, and had lots to do with Scotty in those days, when we were transplanting people we knew had hepatitis B related liver failure, but we had no way of stopping hepatitis coming back. And so we were losing people very quickly after liver transplant from recurrent hepatitis B at that point. And it really wasn't until the mid 1990s and later that we started to get access to very, very early treatments for hepatitis B that we could use in that population, and then obviously more generally. And that's made a huge difference since then.
Yeah, that's amazing. That it wasn't so long ago that we just couldn't do it. Almost sounds medieval, we just couldn't do anything, and everything was sort of supportive. Scott, you were working in a lab at this time, is that right?
Yes. Unfortunately, I predate Simon as well.
What was it like handling these samples that might have these incurable and untreatable infections? Was it scary?
It wasn't scary. What it really gave you, I felt, was some impetus, because it was translational research. So when you've been doing basic research, the end point of what you're doing, in terms of the benefit to people, can be a long way off. But here we were doing something that had an immediate effect. And under Stephen Loccanini, who came back to start up a group, we started looking at hepatitis B using a model, and the model was ducks. So interestingly, ducks have a hepatitis B which can't infect people. In fact, it doesn't do any damage to the ducks at all. But they were a very good model, and Stephen was really looking at what's known as the Hep B mini chromosome that's in the nucleus of liver cells, and also looking at therapies to try and counteract it. And so we started off treating the ducks with some of the new drugs. The first one was one called pencyclovir, which is called Famvir, and is used to treat herpes infections, particularly shingles, varicella zoster, and it had a modest effect. So with Simon's, who are the liver transplant unit in New South Wales, and with Peter Angus, who was looking after the liver transplant unit in Melbourne, we started to give some of the patients who really had no other choice pencyclovir, and we found it had a very modest effect on hepatitis B DNA, again, using quite crude techniques at that time. And then came along some drugs that were originally developed for HIV therapy. So HIV did have a silver lining in that a lot of work went into developing specific antiviral drugs. And I think you're going to talk about this later, maybe, Thomas, but there are some, some drugs that worked on HIV, and HIV and hepatitis B share some common multiplication steps of their genetic material. So first we had lamivudine, and then, so the patients who were on penicillin went on to lamivudine, and it had a dramatic decrease in the hepatitis B DNA, improved survival. And then the trouble is with lamivudine, they started to develop resistance. And then just in the right time, on came another drug called adefovir. And also this went into patients. And then the entecavirs and the tenovirs and the other drugs came along. So there are people from that time, from way back in the mid-90s, who are still around today, who had recurrent hepatitis B from their transplants, that are still walking around. And can there be any greater benefit to like someone working in medical research than to see that sort of outcome?
Yeah. So that's B. Heidi, I want to talk about C, but I guess during this time, this 1986, it's hard to talk about it because it hadn't been discovered yet. That only happened in, I guess, 1989. Can you tell us about when and how Hep C was discovered?
Yeah, sure, Thomas. So hepatitis C was originally referred to as non-A, non-B hepatitis because people who, it wasn't explained as a hepatitis A or hepatitis B related infection, and it wasn't clear what was causing viral hepatitis in this other group of patients. People knew it was a virus because they could filter it, and yet it couldn't be directly cultured from people in the traditional way that we'd isolate viruses in cell lines. And we did know, however, and this might disturb some people, that it did infect chimpanzees, but that was actually a really critical piece of knowledge that actually helped in the discovery of the virus. So in 1989, Michael Houghton was working at Chiron Corporation, and he had been trying for several years to isolate the genetic material of the virus and had no luck. And it was someone in the lab next door who suggested that maybe he should look for RNA, not DNA. And so he used the reverse transcriptase, which Scott was referring to before as being one of the targets of both HIV and hepatitis B antivirals, and used that reverse transcriptase enzyme to copy the RNA back into a DNA form and clone that into a vector, which allowed him to actually discover sequences that were different from hepatitis A and hepatitis B. And he expressed some of the proteins of hepatitis C and found that patients with this non-A, non-B form of hepatitis actually recognised those proteins made from those pieces of the virus of hepatitis C. And that really was the discovery of hepatitis C back in 1989. And then it was still not the end of the story, really, because hepatitis C is actually remains, in fact, almost impossible to grow in cell culture. And so that was such a huge limitation for all aspects of the study of hepatitis C. And then Charlie Rice came along and actually made the first full length, what we call the full sequence of hepatitis C, an infectious clone, and was able to show that that infected a chimpanzee. And then it was some years later, it wasn't until about 2006, I think, that the first virus of hepatitis C was actually cultured in a cell line. So it really has been a very long time, but along the way, there were some really significant advances, not just for hepatitis C, but generally for how we study viruses. And even if we can't replicate them, we could still discover the direct acting antivirals that we're using today were actually discovered. All the research on those had started before we could even grow the virus in cell culture using really interesting methods of studying replication of the genetic material. So it's been a really long trajectory, I guess, for hepatitis C, but once it got to the point where people were able to find systems in which to screen drugs that directly acted on the virus, that actually really quickly led to the discovery of the direct-acting antivirals and then their passage through clinical trials and use ultimately in people with the first drugs in 2013. And they're curative. So this is, I guess, the distinction now between hepatitis C and hepatitis B, that if you take therapy for hepatitis C, you are actually cured of the virus. There's no ongoing residual virus in your body after you've been through direct-acting antiviral therapy.
Yeah, I think it's really interesting that such huge amounts of impact could come from really basic research, just tinkering around with viruses to figure out what's happening. I think one of the things I want to focus on is reverse transcription, because it's quite an important point, and the discovery of that was from basic research as well. In fact, something that didn't seem to have any translational appeal at all. So this comes from the concept of, okay, you read in the textbook, things go from DNA makes RNA, RNA makes protein. This is, I guess, what a lot of people know from their high school biology, but and that was our understanding for a long time, right? But in the 50s and 60s, we actually found some viruses that could turn RNA into DNA. And that, I guess, was really crucial in trying to find Hep C, as you said, because that's an RNA virus and we need to convert it into DNA so that we can manipulate it a lot more easily. So I think I just want to talk about that for a bit, because that process of going from RNA to DNA was a major finding, because a lot of viruses actually did that. HIV being a quite important one at the time, which was just discovered in 1983, just before I was born. So because reverse transcription was so weird, because our bodies don't go from RNA to DNA very easily, it's something weird that we can target. A lot of medicines we have side effects because it affects things in people, but this was only present in viruses. So we can develop drugs against it without worrying too much that we'd have any side effects. And because of that panic around HIV and eventually the successful lobbying of governments to do research on this, a lot of drugs that Scott mentioned were discovered and designed and that could inhibit this reverse transcription process. And we're still using this class of drugs today. And this is where the story sort of converges with hepatitis B, because hepatitis B also uses this reverse transcription to make more of itself. But even though it was discovered much earlier and affected more people than HIV, there was little appetite in funding research for it, and there's still not much. This is important because drugs are really expensive to develop, and you need a lot of basic research to get those tools to work. So what the Hep B field ended up doing is taking these drugs to treat that are being used to treat HIV to see if they work. And a lot of them did, and still to this day, there's probably not one drug that's used to treat Hep B that was originally developed for Hep B. They're all hand-me-downs from other fields, in particular the HIV field. And I think it's important to think about that and think about how much we could have achieved if we focused on B and its unique ways, rather than just trying HIV drugs. And we're seeing just now a lot more of these hepatitis B specific drugs only come out now, more than 50 years after discovering it. So Simone, back when these drugs first came out and were shown to be effective, so this is around the 90s, when we finally had something that worked against hepatitis B, how did that change things in the clinic?
Well, I mean, as I'm a liver transplanter, so we first started using these drugs really in the patients with the more advanced disease. And we, pre-transplant and post-transplant, and to stop the recurrence of hepatitis B after transplant. And it was revolutionary. We were starting to see people who already had liver failure from hepatitis B go back and recompensate. So that means, so when you've got liver failure, you've got fluid retention, jaundice, and we're yellow, blood doesn't clot, really sick, malnourished. And we were seeing people, when we started treating them, reverse and go back to looking healthy again. And that made a really dramatic impact. And then we started using them in people who had had transplants and reversing their significant liver failure after transplant, and protecting them against getting reactivation of hepatitis B. But in the early days, as Scotty already mentioned, the big problem was the emergence of resistance, because the first drug we really had on a big scale was lamivudine. So we started using that in the more advanced disease, and more we started using it in earlier stage disease. And it was very effective at suppressing hepatitis B, but the resistance to lamivudine was really very, very high, particularly in people who had high levels of the virus to start with. And then what we started to see were people starting to have flares of hepatitis. So as the hepatitis got resistant to lamivudine, they'd get a recurrence. The viral load would start going up, and then they'll be having flares. So having severe hepatitis presentations and then going into liver failure because of the flares. And fortunately, and again, Scotty's already alluded to this, just at the right time, we had access to adefovir, and we started switching people. Then we learned that actually it was better to add it on, and we had people on combination. But then they started getting resistance if we just switched them to the other drug. So if they went from lamivudine to adefovir, they'd get resistance to that, and then they'd have another flare. And then we learned to put them drugs together. And then fortunately, we started to get access to tenofovir and entecavir that really didn't have those resistance problems. And that's made the really dramatic difference, so that now we know that if we put somebody on an effective antiviral for hepatitis B, whatever their stage of disease, it will control their virus, and their liver disease will get healthier again, as long as they keep taking it. So now the only weakness in that is access to treatment and staying on treatment, and the patients knowing that they need to stay on treatment long term and having that access. And that access is variable around the world. It's expensive therapy. And I know there's many people who have joined our Zoom meeting tonight who are from other countries, not Australia. So in Australia, these treatments are, very, very low cost to patients, and they can be continued long term. But in many parts of the world, they're expensive and they can't be continued long term. And then there's the risk of hepatitis flaring once you come off.
Heidi, I want to go to you, because with hepatitis C, we're really still stuck in terms of the approaches we have for hepatitis C. The approach we had used for hepatitis B doesn't work for hepatitis C because it doesn't have that reverse transcription step. And those breakthroughs for hepatitis C only became part of it during the early 2000s. Could you give me an overview of, how exciting that research going from the beginnings to now?
Yeah, sure. So because we couldn't culture virus in tissue culture, we couldn't screen drugs very easily that would stop virus replication. So what Ralph Bartenschlager and his team of scientists did was really break up the genome of the virus into the part of the genome that replicates the RNA away from the part of the genome that puts all the surface proteins on the virus, if you like. So we call these subgenomic replicons. And what they are basically self-replicating units of RNA. But it contains all of the, what we call non-structural proteins of the virus. And because they're self-replicating, it gives you a method where you can actually screen drugs that block that replication of the viral RNA. So that discovery was absolutely pivotal to discovering all of the drugs that we're using now in the clinic for hepatitis C. And there was a lot of investment from multiple different companies. There were hundreds of different drugs in development at that time, and it was a real race to see which one would be approved first in use. It also gave researchers a way of understanding how resistance would come up. So we've heard a lot of discussion about resistance with hepatitis B, but a lot of those subgenomic replicons were also used to select viruses that could become resistant to those direct-acting antivirals. And it allowed us to know exactly where that drug targeted on the virus itself. So that gave us confidence that the drugs we were using were targeting specific viral proteins, but it also allowed us to understand how resistance would actually come up. And that taught people that you couldn't just use one drug on its own. You actually had to combine them. And when you combined them, you made it very, very, very hard for the virus to develop resistance. And so that's why all of the therapies that are in the clinic today have 2 or 3 different drugs in combination, because they make it almost impossible for the virus to develop resistance. And that's really key for hepatitis C. That means that, we're seeing very little drug-resistant virus in the clinic now. We've now got what we call perfectivir, where we've got like an ideal combination of drugs that is, more than 99% effective at curing hepatitis C after 8 to 12 weeks of therapy in most people. They work against all of the different genotypes of hepatitis C. And it really is one of the most amazing achievements in science, I think. And quite rightly, Harvey Alter, Michael Houghton and Charlie Rice won the Nobel Prize in medicine for their discovery of hepatitis C, but also Ralph Bartenschlager did win the Lasker Prize for his discovery of the replicon system for discovering those direct-acting antivirals. So I actually think this is one of the greatest achievements in medicine, to be able to actually cure someone of their hepatitis C, and has had a profound impact on people's lives around the world and will continue to do so. Where I think the problems are for hepatitis C in the future is finding, as Jennifer said, all of those people who don't realise they've got hepatitis C and getting them onto treatment. And that's really where I think a lot of the the problems are. Like, we've sort of treated all the easy to find people who are already, seeing specialists for treatment. They've all been treated. Then we've made a few more inroads in people who go to safe injecting rooms. Perhaps they can get treatment now. But then there are all these people out there who don't even realise they've got hepatitis C. How do we find them and how do we get them on treatment? And that's, I guess, the missing millions that people talk about, right?
Yeah. That's right. Now, I guess there's a lot of access issue, as Simone had pointed out, in particular the areas that need it the most. I'm going to cut this discussion short and start to answer a lot of the questions that are out there, I think, because that's really what I wanted this discussion to lead to. Chaturaka, can you load up some questions for the panel, please?
Sure, I can, Thomas. So thank you very much. So I think we have Genoveva, who's got his hands up. I think he wants to ask a question. So the floor is yours. If you can unmute your mic and let me unmute.
Yeah.
I think the participant has to unmute himself. I can't do it from here.
Maybe I'll just make a co-host so that they can unmute themselves.
Are you hear me?
Yes, we can. Yes. Please ask your
Yeah,
question.
thank you. I have a simple question. I'm from Roma ethnic community from Bulgaria. And about me, it's very hard to receive medical treatment from last 10 years or more, because this is very expensive. And we have not some screening program who can adopt my community. And for me, it's very expensive to go to the hospital and to receive medical treatment. So, and I pass it day by day. And for me, we should discuss it is possibly to find some solution, some new programs to adopt minority women from all the world globally who cannot pay for medical treatment and receive tenofovir,
I think this is an incredibly important issue, the access to medications for the people who need them. Simon, can you tell me,
Well,
what's...
Yeah, so I made reference to it. So with hepatitis B, the access has to be obviously complete and global, but it also has to be sustained with our current therapies. And that's as I mentioned, the weakness of our current therapies is that they're highly effective, but you've got to keep taking them. And there is significant risks in suddenly stopping, particularly if somebody's got already quite advanced disease. Stopping and starting treatment is actually, in some people, more dangerous than never starting in the first place. And that's because many people living with hepatitis B won't suffer the complications that we've alluded to before from having hepatitis B. And part of how we assess people clinically is to see whether or not they're in a phase of disease that Thomas mentioned earlier, that they actually need treatment. So not everybody with hepatitis B needs treatment. And in fact, the vast majority of people don't need treatment. It's only probably about 20% or so who do. And the others harm can be done, in fact, if you stop and start treatment in that situation. So it's not just that everybody with hepatitis B around the world needs to have treatment. Everybody around the world with hepatitis B needs to be diagnosed. They need to be risk assessed. They need to have access to treatment. And I totally understand that where you are is treatment is not affordable at the moment, and there are global programs to try and assist that, but they're not focused on your community. I know that. And we also need to have access to identification of the complications of hepatitis B, and that's particularly liver cancer, which is still one of the leading cancers around the world related to hepatitis B. So it's horrible that it's so patchy around the world, and I don't have answers to the local politics, unfortunately.
I want to mention something. Okay, look at me. If I pass it, you don't know me. It's okay. For you, maybe this is not important because you don't know me. You're not my friend. So anyway, but I'm a human being, and like a human being, I should have access to the health care. And I never stop to ask you and local politic and global politic about this problem, because like a human being, I should receive medical treatment. This is my human rights. And that's why I'm here to discuss with you. This is serious problem, because not only Roma community have this problem. The local community in Australia, Aboriginal, in the all the planet, they have minority community. This is the first. The second, this can be structural racism about human rights to access to the medical treatment. Well, I say this with my respect to all of you, like good doctors, but we should discuss, and this is part from medical treatment to the hepatitis B. What's all the best, all of you.
Thank you for that that statement and mentioning this really important issue. I want to talk to Jen about what are the sort of global trends. Are people in these communities getting more access to care over time? Is that getting better?
So I think one of the most important things is that it's just so hugely variable from area to area. And in some places in the world, access has been improving in amazing ways. And there's some countries that have been just doing incredible programs to increase hepatitis B treatment. You know, Mongolia is one example. They've got a sort of nationwide hepatitis B and C registry, and they're enrolling large numbers of people in care, and that's great to see. But we know that globally, less than 3% of people with hepatitis B are even enrolled in care, much less accessing treatment. And so it is increasing on average, but it's increasing much, much too slowly. And there's those disparities that you mentioned still exist, and people who are already at increased risk of hepatitis B, people who have existing healthcare disparities, and as has been mentioned, people who are socioeconomically disadvantaged or who are from First Nations communities across the world, those access disparities tend to exist. So there are definitely good news stories. There's places that have done amazing work in increasing treatment access and that sort of thing. And that's, you know, there's sort of models that we can look to and really see what can be done, even in low income settings, even in places where hepatitis B is incredibly common. But they need to be expanded. They need to be taken to other places in the world to make sure that everyone has access.
Thanks, Jennifer. So I'm going to take a question from Slido as the next question, because those questions have been up there for a few days now. So the first question is related to hepatitis B, what we've been discussing. So we talked about suppressing the viral replication, and Simone quite correctly mentioned that you need to keep taking the drug. So there's an important question up there saying, when will we see a cure? Will we see it in the next 5 years, or how close are we to a full cure?
Thomas, you can answer.
I can answer. So in the Hep B field, we're really, as I mentioned earlier, we're only really starting to see drugs developing now that specifically look and target hepatitis B itself, are designed for hepatitis B rather than, you know, just taken from treatments of other viruses such as HIV or herpes viruses. So I think we haven't been as close as we are right now to some sort of cure. I can't say that it will happen within 5 or 10 years, because these trials are still ongoing. Simone indeed is organizing a lot of them. And I think but the sense is that we probably will have something that, at least in a subpopulation of people, will induce some functional cure. That is to say that you can take this drug for a limited amount of time, and afterwards you won't have to take anything again, and the virus is completely suppressed, and you can live your normal life. I think that will happen. We will have something within the next 5-10 years. Now, how effective that'll be, how big that subpopulation is, we don't know yet. But this is the way, I guess, we develop drugs. The same thing with hepatitis C. In those early days, we didn't have completely effective drugs. They all of those medications went through an evolution and optimization and improvements over that time. And I think that's the sort of thing that will happen with these hepatitis B cures that are coming out.
Thomas, can I add to that, yes, Thomas? So it's so difficult to get a cure for hepatitis B that the top scientists in the field and doctors in the field have said, well, there's actually 3 different cures for hepatitis B. There's a sterilizing cure where you have complete cure. And as you alluded to, Thomas, there's a functional cure where you've actually suppressed the virus so you can't detect it. You can't find the surface antigen, the critical protein in the blood. So they've got these 2 different definitions. The only sterilizing cure I can recollect is the one that the transplant units do. And as I say, the New South Wales and the Melbourne transplant units pioneered this, but took a long time to get it published, as that is pre-treat people with these nucleoside analogues, the entecavir, tenofovir, reduce the virus so you can't detect it anymore. Then you replace the liver. When the new liver comes in, you leave the drugs on for a while, and that way you actually get a cure. You get a true sterilising cure, but that's the only case. But otherwise, we're really punting for the functional cure, and that's because of this little bit of hepatitis B virus that's in the nucleus of our cells, which we call a mini chromosome. So it's like our chromosome, but teeny weeny, to use a scientific term. And it's sleeping there, and we can use those drugs, and it stops the virus from multiplying. We can't find it in the blood. But as soon as we remove those drugs, bang, the virus takes off again. So that's why we have those different definitions of cure.
I just want to raise a small, or actually big issue as well. When we're thinking about developing drugs, particularly for hepatitis B, these drugs need to have very, very, very low side effects, because, I mean, the medications we have at the moment have very low amounts of side effects, and I mean, you stop disease development, or at least slow it down a lot, if you keep taking them. So that's your gold standard. If you're asking people to take a drug that will make them have side effects, that's a bit less, it's a harder ask. And so what scientists are now, and pharmacy companies are now understanding, is looking at it from the patient's point of view. What will I accept as, a cure, and taking that really into account. So these issues are being thought about in the cure space as well. I just wanted to get that message out there.
Thanks, Thomas and Scott, for your input. So we'll go to the next question. So we have Elton, who wants to ask a question, and if you can make him co-host, so I think he can ask the question. Yeah. Yes, Elton, go ahead.
Yes, unmute.
Yep.
Can you guys hear me?
Yes, we can. Yes.
Hi guys. My name is Elton. I'm from Melbourne. I live in Melbourne, Victoria. Originally, I'm from Albania. If you don't know, it's a small country in southeastern Europe, next to Greece. I find out that I have chronic hepatitis B last year. Just by surprise. I don't know how I have it. And I've been for the last year in entecavir. I just wanted to ask you, long-term like side effects, like say for the next 20 years, if there's no cure. So what are the side effects? Like, I just want some answers on like side effects.
Thanks, Elton. So I think that's the question for Simone.
I think so.
Yeah.
So we've got two very, very effective drugs currently in Australia. It's a bit different around the world, but entecavir, which you've been placed on, and the other one is tenofovir. And as far as we know at the moment, with entecavir, we have no significant side effects, either short-term or long-term. So we're not seeing, we've had patients on entecavir now for a long time. We've got lots of experience in using entecavir in chronic hepatitis B. It's highly effective. It prevents liver cancer, it prevents liver failure, and it reverses liver failure. And it doesn't have any long-term effects on any of your other organs. With tenofovir, we do have some concerns in some people. It's rare, but some people who are on tenofovir rather than entecavir have some impacts on kidney function and on bones over time. And that's why there's a new formulation of tenofovir that's been developed that is available in some parts of the world and is used in HIV medicine because it doesn't have those effects. But for entecavir, which is the drug that we tend to use the most, the reason we do that is because it's highly effective and doesn't have any long-term side effects. So I'd be very reassured that you're on a great drug that's highly effective. It's going to reduce the risk of long-term effects of hepatitis B significantly. I'll never say completely, but significantly, without any long-term consequences. Again, you've got to keep taking it unless your doctor that's prescribing it says that it's okay to stop it. And we are looking at some strategies in some people, once they've been on entecavir or tenofovir for a period of time, to look to see if we can stop it and maintain good viral suppression without effects of reactivation of the hepatitis. But those strategies aren't commonly used in the clinics at the moment. So I would recommend anyone who's been prescribed to stay on long term unless their specialist says it's okay to stop because they've reached some sort of endpoint.
Thanks, Simone. So I think that was a very comprehensive answer.
Can I
sure, Thomas. Yeah, go ahead.
Say, Elton, that it's really. I'm talking as someone who has been on long-term antiviral therapy myself. I have hepatitis B, so I've been taking medication for years, and it is normal to have these fears because a lot of the pages on the internet, you might try to look up information. They have all of these side effects, and it's really scary. And I feel what has helped me is talking to other people who have been on long-term therapy and they've been living fine with it. So I'm happy to talk to you or chat with you if you want. We have a forum for people living with hepatitis B. I'll put the URL in the chat. Contact me there, register. There's a whole community there. We're about 400 to 500 people. So, so anyone in this Zoom chat, please, if you have any worries or any follow-up questions after this, please just come to the forum.
All right. Thank you, Thomas.
Thanks, Elton. So next I will take a question from Slido and I'll come back to Jimmy after that. So there's a question, is there a correlation between hepatitis B and having low lymphocytes? Let me rephrase that question. What about immunosuppressed people, and staying on this topic of hepatitis B, are they more likely to get hepatitis B or once they get hepatitis B, do they have more adverse outcomes than people who are not immunosuppressed?
Guess that's for me as well. So being immunosuppressed with hepatitis B tends to put the hepatitis B level up, so the viral load up higher. And that's because the way hepatitis B is controlled is by having an intact immune system suppressing the virus. So if you suppress the immune system, the virus can get higher. And so we see that particularly an issue. There's a form that's a very interesting form. And again, we've worked with Scotty many years ago on this, and unfortunately don't see it anymore, that in liver transplant patients or in bone marrow transplantation or kidney transplant patients where they are immunosuppressed, the level of virus gets to such a high level that it actually overwhelms the hepatocytes, the liver cells where it's replicating. And those liver cells swell up and balloon and stop working. And we only really see that disease in people who are immunosuppressed. And we've also seen in hepatitis C in immunosuppressed people, again, the same sort of mechanism where the virus is allowed to sort of replicate to such a high level, it overwhelms the function of the liver cell that it's growing in. Doesn't quite get to the why lymphocytes can be a bit low, but it's about the interaction with the immune system, which is absolutely critical in both hepatitis B and hepatitis C. Lymphocytes, yeah, we see in people with active viral diseases.
Oh.
I think we have a problem with the internet connection.
I think Simone's
hopefully Simone will get back.
has been locked down with.
Hopefully Simone will get back soon. So in the meantime, so Jimmy has his hands up. So if you can ask your question. Jimmy, you are co-host now, so you can unmute yourself and speak.
whether I'm loud and clear. Can you hear me?
Yes, we can.
Okay. Okay. Yeah. Thank you. Thank you so much, guys, for organizing this event, this Zoom chat. My name is Amone Jimmy. I'm all the way from northern Uganda in Lamuo. I am also actually a Hep B patient. I got to know about the virus in 2016, and it was a bit scary, though. Well, the question that I have is, at what point do someone get to start taking medication to like suppress the virus? I don't know at what point exactly. And then, I'm not a very technical person when it comes to this medical field. And then also within this kind of community, because in Northern Uganda, the percentage of people living with Hep B is quite high, although most of them sometimes is not well documented, but I live in the community. I get to see this, many people always, coming open and talking about this. So the type of medications that it's usually recommended that we can let the people know. I don't know which kind of exact medication. Sorry, I came in a bit late. If this was already answered, you forgive me. And then the final question is, just like one of the, I listened to one of the speakers, I didn't get the name quite well. She asked a question about how the Hep B is not being, recognized as one of the, the diseases that is a threat to life. In terms of when we seek for support or funding, because the treatment here is really quite expensive, let alone the treatment, even the scanning, just to go and do the test and go for further screening, maybe to get the virus load. It is quite expensive. Most people end up just doing the first test, the initial test. And then when it now supposed to go for viral load, it becomes so expensive. Most people now just assume. And that's why I asked the question, at what point do you think someone can get to know when they start the treatment? They can start the treatment. So it's really quite a challenging situation down here for us. I know it's not only here, but I've started coming up with some CBO, community-based organization, to support the communities. One is with sensitization, so that people get to know the basics about hepatitis B, because a lot of people do not know the basics. You might find somebody could be saved by just giving them information, and maybe they stop consuming alcohol. But if they don't know, they will continue with all sorts of lifestyles, not knowing that they're endangering the liver as a result, as a added result of hepatitis B as well. So it is something that I think we also, the question is, we need to really support, we need to out there look for those who are willing to support, like community-based organization or other organizations who are thriving, more especially for me, to be in this, really go out there and start sensitizing. Because for me, I'm happy to, I was blessed that I knew something to do with computers and internet. So I started Googling more about hepatitis B. So I got into become self-aware and what I needed to start doing, like good feeding, eating a balanced diet, proper diet, and staying away from alcohol. But people in the no information, so I want to come up that I would share about this hepatitis in the Yes, please.
Yeah, I think your audio was breaking up, but I think we've got the gist of the question. Simon, I guess. Oh, sorry, I'm taking it from Chaturaka. But I guess, Simon, the thing about treatment eligibility and when we choose to treat people, I guess.
Yeah, sorry if I dropped out before.
Oh. I think she's dropped out.
Sorry,
but
sorry, I'm back
she's
again.
back, yeah.
I've tethered to my phone. My Wi-Fi keeps dropping out. So I'm a bit unstable. So sorry. Yeah, sorry. Yeah, in a country like we live in, many of us, we do have access to regular testing, so that we can fully assess somebody living with hepatitis B. And what we do look at is what the level of the virus is, how it's affecting the liver, how much scar tissue is formed in the liver, and then we make a decision then about whether they should be having treatment. Now, a lot of those assessments are not routinely available in many parts of the world. And the treatments, as you mentioned, are expensive and inaccessible. And therefore, we do rely on lifestyle modification, and particularly alcohol would be the major factor that can drive liver disease in that situation. But we do need a global approach to access to therapies, and treatments have to be affordable wherever you live around the world. And the assessment for the liver needs to be affordable and available as well.
Simon, do you think there are some good surrogates we can use, like hepatitis B, E antigen, and liver function tests, and APRI scores from blood tests? Those sorts of things might be helpful.
Well, again, you still need access to the blood test in the first place to do those. So those tests
are
a lot cheaper than doing an RNA level. Yeah. Certainly, we do know that if somebody's got E antigen, that they've got a very high level of virus. So yeah, you're right. We can that, but even then, in younger people with a high level of virus, we don't necessarily have to treat at that stage because it doesn't really benefit them in the medium term, as far as we know, until their liver disease starts and they're starting to get abnormalities of liver enzymes and liver function. So just having a high level of virus doesn't mean you need to treatment either. At this stage, it may change over time, but that in our current guidelines around the world, they're not the people either. It's really the people where the liver and the immune system is starting to react against the virus, that they're starting to get liver injury, that then we need to target to suppress the virus. But again, it is challenging for many, many parts of the world.
Thanks, Simon. Yes.
Oh, sorry, Scott.
Yes, Thomas.
Is there research going on in making diagnostics cheaper or more available or easier to?
Obviously, we started off just doing in-house, and that can be very, very inexpensive. But I guess the drawback is the lack of reproducibility, and it's like one place, and how do you work out? You need standards to sort of determine what level of the virus is. The tests themselves aren't super expensive, except for the cut that the manufacturers are making, really. But I don't see how we can get around that. There are lots of effort going into supplying point of care molecular tests, and again, you'd be aware of these. And other countries, whether it's non-government organisations or through WHO, trying to supply these tests at a much cheaper rate, but still it may be too expensive in low and some middle-income countries.
Thanks, everyone. So I'd like to pivot to hepatitis C for the next question, and next one is therefore to Heidi. So we have very effective vaccine for hepatitis B, but not so great treatment. And it's the reverse actually for Hep C. We have great treatment, but not a vaccine. So Heidi, do we need a vaccine, and where are we in terms of a vaccine for hepatitis C?
Yeah, it's a great question, actually, and a really interesting contrast between hepatitis B and hepatitis C, I think. I think, for a long time, I think one of the other considerations in why we haven't gotten further with Hep B treatments is that there was a hepatitis B vaccine. And there was a big, sort of in my early career stage funding for people doing basic hepatitis B research really collapsed because there was a hepatitis B vaccine. And so people would say, well, we've got a vaccine, why don't you just prevent infection and then you don't need treatments anymore? And I think the reverse is actually true for hepatitis C. The incredible success we've seen with DAAs has also seen the funding become really hard to get for hepatitis C, because people say, well, what do you need a vaccine for if you've got great treatments? Shouldn't you just invest that money in treating more people rather than investing that money in developing a vaccine? And, you know, I have no black and white answer for this, except, you know, in an ideal world, you'd want to do both. You'd want to do everything. You'd want to be doing harm reduction, education, cheaper treatments, better diagnostics, and you'd want a vaccine. But we know that's not how it works in the real world. And there's always priorities and arguments to be made one way or the other. I think for hepatitis C, where we are with vaccines is we've got a lot of really great candidates at the moment. And where we're finding it very difficult to progress Hep C vaccine development is actually doing, well, A, getting the funding to do the clinical trials, and B, finding populations that we can do clinical trials in typical high-income countries, because the incidence of hepatitis C has gone down so much. It's actually becoming really difficult because you have to have really huge trials to get enough incidence of new infection to actually evaluate whether your hepatitis C vaccine is preventing infection or not. The other really complicated thing about hepatitis C is that we now consider that the vaccine doesn't necessarily have to prevent infection. It has to prevent chronic disease. We accept that even with the best vaccine, you may get some infection, but what we don't want people to progress to is that chronic form of hepatitis C. So that means you've got a very long window where you're monitoring people after they've been vaccinated and they've been exposed to virus to actually see if they develop chronic infection. So that's a bit more complicated to how we'd normally just study efficacy in a vaccine trial. So there's a lot of debate going on at the moment in the viral hepatitis C community about whether we should consider what we call a human challenge model for hepatitis C, where we deliberately infect vaccinated people with hepatitis C because we know we can cure them. And so this would be a way of using fewer people to determine if our vaccines are actually effective. And it's a really interesting debate because there's ethical considerations, health considerations, you know, which virus do you use? Because there's so many different genotypes of hepatitis C that you could pick for this. You know, how long do you have to study people for? And what are the long-term effects of deliberately infecting someone with hepatitis C? Even if you can cure them, they may have hepatitis C for 6 or 9 months before you actually put them on therapy. And that could have long-term impacts that we don't yet understand on their, you know, their whole bodies. So it's a really interesting space at the moment for hepatitis C vaccines. And there's a lot of movement now of people who used to do all the work on the direct-acting antivirals has sort of moved into the vaccine space now. And we've got actually quite a large consortium of people coming together globally to work together to solve some of these questions. But there's no, like, you know, I couldn't tell you that there was going to be a hepatitis C vaccine in 5 or 10 years because there's nothing really there at the moment that you could say is looking promising even. The last vaccine trial that was done was a phase 2 trial of what we call a T-cell vaccine, and that failed to show efficacy to prevent chronic infection. There was some indication that viral loads were slightly lower in the people who got the vaccine, but it wasn't sufficient to actually show a difference between the group that didn't get the vaccine.
Thanks, Heidi. So that was a comprehensive answer. And another important fact to consider here is that though we have effective treatment, it doesn't prevent reinfection. And in some parts of the world, the treatment for Hep C also is really expensive. We are lucky in this regard for Australia because it's free for people
That's
under
right.
Medicare, but not so for other parts of the world.
Absolutely.
So I'm going to take another question going back to hepatitis B on the chat. So the question is, what are the treatment options for a patient who has no detectable DNA, but was positive for surface antigen?
Probably, there's some work going on at the moment, which Simon was talking about before, whereby patients who have got undetectable DNA and their surface antigen levels low in the blood, because you can quantify that, that if they go off therapy rather, they usually get an immune flare, so their liver function goes up, but a small proportion of those people actually get a cure. Again, someone mentioned this before, whereby a functional cure, whereby the surface antigen disappears, maybe with or without development of surface antibody. But the trouble is, at the moment, that's probably only 1 in 20 or 1 in 30. So again, the researchers are looking at, are there other predictors, are there other biomarkers that we can use to improve that maybe up to 10%? And then you might have a 1 in 10 chance of having a functional cure. Simone, you might want to comment.
Well, I was the add-on to that is some of these new therapies that Thomas was alluding to before that drive down hepatitis B surface antigen are initially being actually investigated exactly in that population, people who are on long-term antivirals, I think that's what you're talking about, who are suppressed. The virus is undetectable, but they're still surface antigen positive. And those new agents are looking to see if we can then flick and clear that hepatitis B surface antigen through those approaches with a short course finite therapy. It's going to be pretty expensive, at least initially, but whether or not we can convert people into that functional cure that Scott was talking about. So the different approaches. So one is just to stop the therapy altogether and let the immune system do it. And the other is to use some of these novel agents that are targeted at the hepatitis B surface antigen and drive that down in that setting. So they're really interesting approaches, but at the moment, there's nothing that we can recommend in the clinic to do that.
Thanks, Simone. So next up, we have a question in the, not a question really, a comment from Kathy Jackson in the chat box saying that we also need to consider treatment to reduce transmission, for example, when pregnant with a high hepatitis B viral load. Any comments?
Yeah, I might. Should I take that one?
Yeah, sure.
As well. So it's a global approach to do that. So WHO has made very clear statements and has specific guidelines about treating women in pregnancy with particularly high levels of hepatitis B to prevent mother-to-baby transmission. So we know for many women with hepatitis B, they don't have a high level of virus, but they will still transmit to their infant. But if we give that infant vaccination, and particularly in combination with passive immunity, which is providing antibodies within 4 hours or maybe 12 hours of birth, that we can prevent hepatitis B in that baby. But in women who have got very high levels of virus, even if we do those things, we give the baby vaccination and immunoglobulin, that about 8.5 to 10% of those infants will still get hepatitis B in that setting. And so in many parts of the world, and now are really being promoted by the WHO, is to use an antiviral, and specifically tenofovir, because we know it's safe, use that in the last trimester of pregnancy, and that's almost universally effective at preventing transmission to the baby. So it's an incredibly important strategy globally, while we still have people living with hepatitis B, to protect the next generation, because we know around the world that most people who have hepatitis B got it at the time of birth. And this is really to protect the next generation, and a really, really important strategy, because it's going to still take a while for global vaccination of populations to prevent hepatitis B in everybody.
Thanks, Simon. And to add something regarding that on hepatitis C space. So treatment as prevention is now also gathering momentum in hepatitis C space also, because recently a trial was concluded and published by Kirby researchers in New South Wales prisons, where treatment as prevention in New South Wales prisons showed that there could be a dramatic reduction in new infections for hepatitis C as well. So that is again something that is gathering momentum. Yes, Thomas?
Can I ask Jen whether she has sees evidence of treatment as prevention of transmission for B?
For B, I don't think so. I was just going to mention that on Hep C, we've actually seen a 90% reduction in new infections for hepatitis C in Victoria, specifically since 2016, which is really incredible, and I think speaks to that point about treatment as prevention. For hepatitis B, it's a little bit trickier to measure, specifically for prevention in pregnancy. There have been some studies that have shown this really effectively, like someone said, the evidence about the reduction in risk of transmission for women who are treated in pregnancy. But more broadly in the community, I think it's just going to be less of an impact because we do have vaccination, which is such a strong tool to protect people from hepatitis B infection. So that's probably the first step that we would take in order to prevent it from being transmitted. But yeah, that's another option. And for some particular people, so particularly people working in medical settings, that's an option for them to be able to work in exposure prone procedures, for example, if they're working with Hep B without the risk of transmission. So it definitely has its uses.
Can I have a follow-up question? Maybe someone can answer this. How, or Jen, how actually common is horizontal transmission of hepatitis B from adult to adult? Do you see it often in the clinic? Does it, bear out, or is really the majority vertical transmission?
Well, globally, the majority is definitely vertical transmission, mother to baby transmission around the time of birth. But we still see unvaccinated adults acquiring hepatitis B through sexual transmission, through injecting drug use, and other percutaneous exposures, tattooing, etc. But it's infrequent as larger numbers of people in the population are vaccinated. And for many countries in the world, there's now universal neonatal vaccination. So that cohort of children will grow up with immunity to hepatitis B. Many countries, such as Australia, had catch-up vaccination for people at the age of 13. So again, that cohort will grow up without being at risk of hepatitis B. But we still have people older than that who miss those vaccination programs, who remain susceptible to hepatitis B around the world, including in Australia, because they've never been vaccinated and they don't have hepatitis B and they remain susceptible. And so every year, again, wearing my liver transplant hat, we still see people with adults acquiring hepatitis B through those routes of transmission because they've never had immunity and they develop hepatitis B. And while most people will get Hep B, maybe get a bit sick and get better again, some people will get liver failure from it and need urgent transplantation if it's available, or die as a consequence of it. So it again breaks my heart to see people with vaccine preventable illness getting that illness, and that goes for hepatitis B, it goes for hepatitis A. I know there was a question on there about hepatitis A, I noticed, but any vaccine preventable illness, seeing people get that illness where they could have been protected is pretty sad.
Yeah, absolutely. And I'll just very briefly mention that in Victoria and in most states in Australia, vaccination is funded by the state health department for household contacts of people living with people with Hep B, people who inject drugs, people who have multiple sexual partners. There's a lot of different indications. And so it's actually something that can be accessed for free. So it'd be great to see that taken up more and prevent those really tragic outcomes.
Thanks, Jen. Unfortunately, I think our time is up. It's 7:30, and we had a very engaging conversation, and thank you for all the panelists. There are a few more questions that we couldn't address. Is there any ways and means, Thomas, that we can answer these questions later offline?
Yes, if the panel is willing to take questions and email them out, perhaps. Since Chaturaka, you have the emailing list for the event, we can try to collate and answer some of those questions. And again, there is hepbcommunity.org for people who are living with hepatitis B or are or affected in some way with hepatitis B, and we're happy to answer questions there as well.
Yes, and so this is on behalf of ACHV, and I'd like to close the meeting now. And this is the first time we organized something like this, and we were not sure that whether there'll be a lot of interest in this in the community. But I think We've seen a lot of registrations and people asking a lot of questions, people joining from all over the world and sharing their views and perceptions, and of course their problems they have in getting access to care. So I think this is really encouraging, at least to keep the discussion going. And I would like to thank all the panelists for their time and their expertise,
Yes.
and joining in. And thanks for all the participants, and thanks everyone. So hopefully we'll see each other again in a similar meeting soon.
Yeah. Thanks, Chaturaka. Thank you, everyone.
Good on you, Thomas.
Thanks, everyone. Thanks, everyone.
See you all.